Hello, students. This video is going to look at some ideas about what species are and also investigate some of the processes by which lineages are able to transform from, say, one ancestral species into multiple descendant species. And so this really kind of the whole question comes up, what is a species anyway? And as it turns out, the concept of a species is sort of like a human construct. Species inherently do exist, of course, but their classification is something that we have determined using a variety of different systems. And some of those systems are, I wouldn't call them arbitrary, but they're based on different sets of criteria. And so, although we like to put things into boxes, sometimes things don't always fit perfectly into boxes, but that is kind of the way that humans think. But as it turns out, there are at the present time 26 different species concepts used. So these are 26 different sets of criteria used to say one species is different than another. So we have things like the biological species concept, the morphological species concept, the phylogenetic species concept, the evolutionary species concept, which has to be the right one, right? Or the ecological species concept is yet another. There's also something called the differential fitness species concept. And maybe you even have a species concept of your own. And so this quickly starts to become sort of overwhelming, like all these different species concepts. Which one's right? Why are there different species concepts? It might not seem to make a whole lot of sense. Why don't we just have one species concept? Well, one of the reasons is because life is so diverse and different taxonomic groups could be classified in different ways according to different criteria, just because of what we would call their life history or sort of the way that they do things is really, really different. And so one species concept might not really apply to every single taxonomic group that exists, as well as others. We won't talk about all 26 concepts, but we will talk briefly about some of the ones that are most commonly used. And probably the original species concept used by naturalists, say like Carl Linnaeus in the 18th century, was something called a morphological species concept. And by this concept, we're basically saying that a species is a group of individuals who look similar to each other, but different from other groups. And this is pretty intuitive because we can go out into nature and see that lots and lots of different living things appear to look very differently. And so it's not surprising that early naturalists and probably even people who were non-scientists before this time were able to classify things as being different species, really just simply according to the fact that they looked different. We can look at a whole different group of different beetles, groups of different beetles, and see lots of variation in their morphologies or their sort of physical characteristics, and use those differences to classify these different beetles into different species. Likewise, we might do something similar with woodpeckers, or we might say, hey, all these woodpeckers look so similar, they must be the same species. Likewise, this kind of concept has been applied to plants. And so many plants might look different, and so we're comfortable calling them different things. And so this system of classification has been used for quite a long time. It probably, again, goes back to the times of folk taxonomy, where folks simply were saying, well, these things look different, or these different plants, for example, have different uses. Let's give them different names, and we're basically going to call them different morphological species. And so this was accepted for a long time until Ernst Mayr came along with a different concept. Because as it turns out, sometimes things that look really, really similar don't necessarily exchange genes with one another. And this leads to the so-called biological species concept, which more specifically gets at whether or not individuals within different populations are breeding with each other. And so basically, by this definition, we say a species is a group of actually or potentially interbreeding populations that are reproductively isolated from other groups. And when we're saying reproductively isolated, that's a fancy term for individuals from two different populations are not exchanging genes. And we can kind of look at this cartoon here that sort of describes what this situation looks like in a spatial context. We have these 7 hypothetical populations, but not all of these populations have gene flow between them. Maybe populations 1, 2, 3, and 4 share genes, populations 4 and 6 share genes, and maybe population 7 doesn't share genes with any of the others. And if this pattern of reproductive isolation goes on for long enough, it really can lead to these different populations being described as different separate species under the biological species concept. And I would just invite you to recognize that for the most part, what you're probably going to see across sort of general biology is when people do talk about species, they're often talking about species that have been designated via this thing called the biological species concept. So this has come to be a lot more widely accepted than the morphological species concept. Now, when we're talking about reproductive isolation, we're talking about certain mechanisms that cause that to happen. And basically, these mechanisms either can operate before individuals attempt to breed, or they can operate after individuals have attempted to breed. And we would call those two different kinds of mechanisms prezygotic or postzygotic. Zygotic referring to the formation of a zygote via the process of fertilization of sperm and egg. And so prezygotic isolating mechanisms might be things like the fact that maybe potential mates do live in the same area, but they never come across each other. They never cross paths, either because they occur in different sections of the habitat, or they're active at different times of the day or different times of the year, for example. Or maybe potential mates do come together and interact, but they don't mate simply because they don't recognize each other as mates, for example. Or maybe individuals do attempt to undergo fertilization or copulation, but the gametes just can't get transferred. The sperm just cannot successfully fertilize the egg. And so these are different ideas of things that can prevent a zygote from ever being formed between two different individuals. Prezygotic isolating mechanisms. Now, in contrast, sometimes, let's say, that fertilization happened, but maybe the zygote's not very, very viable, and so it dies early during the process of embryogenesis. Or maybe these offspring are formed and they survive, but they're inviable and they cannot go on to reproduce themselves. Or maybe they're sterile, or maybe when they do go on to reproduce, their offspring are also sterile. So there are these different mechanisms that are basically preventing reproduction from being successful when certain individuals have the opportunity to potentially mate with each other. And it's pretty obvious that certain species don't seem like they would ever have a chance to mate with one another. Like birds and humans are probably not going to be mating with one another and having offspring. But there are other examples of situations where there are species that are very, very closely related in terms of their evolutionary history that potentially could breed with each other, but they just seem to be posed with these challenges caused by reproductive isolation. And then nowadays, with the advent of a lot more molecular genetics analysis, or that is to say, analyzing DNA sequences, there's yet another species concept that is in use called the phylogenetic species concept. And this concept basically says that a species is the smallest monophyletic group that can be distinguished by some kind of a shared derived character. So, for example, we have these 5 hypothetical species here, and each particular group seems to be united by a particular shared derived character. And we were comfortable calling these different groups separate species because they each share unique derived characters. For example, maybe with these flowers in the cartoon at the bottom, we have 2 different species kind of really being classified based on flower shape and flower color. And so these 2 different clades have very, very different shapes of their flowers. And maybe they also have different colors of their flowers. And so we can use these types of characteristics that are not just based on DNA sequence, but also based on morphological characteristics to distinguish clades as being individual species. But again, this is very frequently done nowadays using DNA sequence, which has a pretty powerful ability to detect relatedness amongst lineages in ways that might not be obvious based on simply just looking at morphological characteristics. And so this comes back to the question of, well, which of these concepts is correct? And once again, to reiterate, these concepts are all human constructs. And so basically, species are being circumscribed into whatever particular classification that they have according to different sets of criteria. And basically, the answer is it really depends. And as it turns out, the species concepts that are going to be applied are usually those that are being chosen by experts in these particular fields. And usually, certain experts will focus on very specific taxonomic groups. So it's not like, there's one biologist who's out there saying all these species are being classified the way they are because they know everything about every single species in the world. That's almost never the way it works. It's more like there are experts in bacteria, there are experts in certain lineages of archaea, there are experts, say, in herpetology, that is the study of reptiles. There might be experts within certain reptile groups. Likewise, within arthropods, there might be certain insect experts who really focus on just one group of insects, maybe even uh, a group of flies, not all flies, but a group of flies, or a group of beetles, or within plants, certain experts who focus on a certain plant family or even a certain plant genus. And they're the ones who are kind of looking at the data and making the call that, hey, we should call these things different species or not. So to also illustrate, when we talk about species, we're usually talking about something that looks like this. And a species is typically described according to the genus that it belongs to, and then it is assigned a species epithet. And then often these species descriptions will be accompanied by the abbreviation of the author or the person who came up with the species name. For example, Carl Linnaeus named our species Homo sapiens, and capital L period is the abbreviation of his name as the author of the species. And so you'll typically see species written in this fashion. And if you notice, species names are always italicized. If they're not italicized, then they'll be underlined. But you will never see someone trying to say this is a species without italicizing it in writing or underlining. And these species names, they're useful in a lot of ways, and that's why we've come up with them, and that's why we're classifying things in these ways. And these species names sometimes can be confused with common names because Homo sapiens could have multiple common names, perhaps, depending on which particular group of humans or culture you're looking at, but we're all one species. But typically, using a species name allows us to communicate broadly across the entire global scientific community about a very specific taxon that we are discussing. So that someone who doesn't speak English, if they read Homo sapiens, they know that they're reading about humans, our species. And this is true of all other kinds of species designations that you might come across. Now, the whole delineation of species really can get complicated, and it can start to lead to this interesting sort of spectrum of variation in species either being kind of lumped together too much or maybe being split apart too much. And so conservation biologists recognize that there kind of needs to be a stable medium by which we are defining species without putting too many different populations into one species or breaking too many populations apart into many species. And this idea was discussed in this paper from Frank et al. In 2012. Basically, the implications of different species concepts for conserving biodiversity turns out to be kind of important. In short, the idea is that we might have these 6 different populations here, A1, A2, A3, A4, B1, and B2. And in this hypothetical example, A4 is a very small threatened population. And there's a question about, well, which species do we assign this to? Do we call all 6 populations members of the same species? Or do we break up A's and B's into separate species? Or should we consider A1, A2, and A3 one species, A4 its separate own individual species, and B1 and B2 yet another species? Why does this matter? Well, this is based on the idea that if individuals from, let's say, A1, A2, A3, A4, B1, and B2 are all able to interbreed with each other, there might be a loss of fitness because the A's and the the A populations and the B populations are a little bit too distantly related from one another in order to produce healthy offspring, and we would call that outbreeding depression. In contrast, if maybe we split the A1, A2, A3 populations into one group and allow them to interbreed, and they appear to produce healthy offspring. And maybe B1 and B2 can be split into a separate species, and they produce healthy offspring. And we decide to split A4 into its own individual population. A4, because it's small, might really suffer from inbreeding depression, probably because of the effects of genetic drift. And so while we might think it's a good idea to call it a separate species, it's really suffering from losses in genetic diversity by the fact that it's not considered an adequate group to be interbreeding with these other two groups. And so this kind of poses a difficult situation for the conservation of population A4. And so in this paper, the idea that's suggested is that really just grouping all of the A populations together as one species and the B populations together as one species seems to be a situation where A4 can benefit from some genetic admixture from A1, A2, and A3 if all four populations are exchanging genes via gene flow. And this allows us to have something we call genetic rescue in the case where we have a threatened population. And so this whole situation kind of really depends upon the number of shared alleles that each of these different populations has, and really what is the fitness of their offspring. If the offspring are fit and able to go on and produce more offspring in subsequent generations, why not call them a species? And this is very, very much like what we saw when we looked at the biological species concept, where we have potentially interbreeding populations, and if they are free from reproductive isolation, then they make good species. And in this concept here takes it a step further by saying if they're free from reproductive isolation and they're producing healthy offspring, then they also do make good species. So this is kind of reinforcing the biological species concept and doing so in the context of conserving threatened populations. And then the second part of our question here was sort of like, how does one species become more than one species? And this is kind of a little bit of a more intricate process to get into thinking about, but basically populations do diagnosably diverge via the process of speciation. And as it turns out, there are a few different avenues we understand that allow this to happen. Such as things like allopatry, which is sort of a fancy word for the geographic separation of different populations, sort of ultimately becoming different species. Something called sympatry, which is sort of like behavioral separation of populations in the same environment, where they're doing different things and they're sort of experiencing reproductive isolation as a result. Things like parapatry, which is kind of a fancy word for semi-geographic separation across space, and this often happens along genetic declines, involving some levels of local adaptation, probably. Things like hybridization, where two species actually make a new species, maybe sort of by accident. And then there's this really crazy process called polyploidization, where one parent kind of is contributing to making a new species really totally by accident because of mistakes that are happening during certain types of cell division that we call meiosis, which is the process of cell division leading to the formation of gametes. So sometimes when there are mistakes in the formation of gametes, it can lead to these very unusual cells forming that can contribute to the formation of new species. So we will review each of these concepts in a little bit more detail. I know it might sound slightly overwhelming at this point in time, but we'll get through it. So again, think about this idea that geographic patterns of gene flow really can affect evolutionary outcomes. And the spatial distribution of different populations really is determining the probability of both this dispersal of individuals among the populations and the probability of these different populations sharing genes, sharing genetic diversity. So again, we might have our 7 hypothetical populations, but not all of them are exchanging genes with one another. And this starts to lead to different patterns in the sort of genetic makeup of each of these different populations. And this can lead to the process of them having some level of reproductive isolation. Yet again, how does this play out in the evolutionary game? Well, as it turns out, the size of the different patches, or the size of the different habitat areas that these populations occupy, and the amount of gene flow between them really, really influences their effective population size, and it really, really influences the effective genetic drift. That is, these random changes in gene frequencies that are occurring within these populations because of how large they are, because of how large the area they occupy is, and because of the probability of them exchanging genes with neighboring populations. And so genetic drift alone can start to drive differences in gene frequencies amongst these different groups, causing them to become a little bit different than one another. At the same time, if there are environmental differences amongst these different patches, that can also drive natural selection to have different kinds of pressures operating on different populations. And so we see this situation where there's probably some level of random genetic change going on that very much depends upon the population size. And then the different geographic areas might vary somewhat in terms of the different types of environmental factors that natural selection is operating upon that might be related to certain adaptations. For example, this group here might possess certain traits that are more suited to these environments, whereas this group here might possess certain traits that are more suited to that environment. And likewise, this group here might possess certain traits that are more suited to this environment. And they're sort of, again, reproductively isolated. And if this sort of process plays out for long enough, and there is some level of restricted gene flow, then we can really get to the point where these 7 different populations have really diverged into 7 populations that make up one species have diverged into 3 separate species because of the processes of random mutation, genetic drift, and variable selection pressures in different environments, also being driven by different patterns in gene flow. So we see our 4 causes of biological evolution operating in the process of speciation: mutations, random genetic drift, natural selection, and gene flow, all playing out to influence how these populations may have become differentiated over time. And this kind of leads us to this first concept we mentioned called allopatric speciation, or allopatry. And this is typically something that occurs when a geographic barrier separates different populations. And this happens via 2 sort of contrasting models that we call vicariance and dispersal. Vicariance is also known as the dumbbell model, and dispersal is also known as the peripheral isolate model. These are kind of fancy terms, but basically we're saying this either could happen when a single population is split into 2 different populations, or when a single population has a small subset of it disperse away and colonize new territory to ultimately diverge, probably genetically. And this probably, if you recall, might look a lot like the founder effect that we discussed when we looked at genetic drift. So in the vicariance model, we have our original population, and this population is all connected, and all the individuals are exchanging genes. But then at some point in time, a geographic barrier to dispersal arises, and it separates this population into 2, or theoretically more, separate groups. And if this goes on for long enough, and these 2 separate groups are not exchanging genes any longer because there's this new barrier to dispersal, which could be something like a mountain range forming, which would take a long time, but this is one way that speciation is known to happen, or a river suddenly flowing through an area, dividing populations in a valley into 2 separate halves, just for example. If this geographic barrier persists for long enough, and the genetic differences on either side of the barrier start to accumulate for long enough periods of time, and the environmental differences also start to add up, where selection's not quite operating in the same way on either of these 2 populations, then we might see a situation where there's very strong reproductive isolation between these 2 different groups. And they will reach a point where, if we did experimentally take individuals from one population or one group here and individuals from this other group over here, and we tried to get them to breed in a laboratory setting, for example, they might not be able to do so. And we would say, if we were using the biological species concept, that they have become two different species, although they did share a common ancestor at one point in time. Something very similar is happening with the dispersal model, where this small subset of the original population colonizes a new area. And again, they're probably accumulating different new mutations. Maybe if it's a small population, also it's really susceptible to the effects of genetic drift because it has kind of small effective population size. And maybe this new region is environmentally very different than the region where the original population occurred. And so the selection pressures here and the selection pressures here are not the same. And this can start to drive, again, reproductive isolation. And we might see a situation where, again, if we tried to take individuals from this group and individuals from this group and get them to breed with each other after all this time has passed, they might not be able to do it. So this is one of the classic ways by which many species have diversified, or many taxonomic lineages have diversified. For example, in California, we have a very diverse flora, a very diverse composition of plants that we have in our state, and a lot of them probably did diversify via this vicariance model. The Galapagos finches, as another example, probably diversified in some ways via the dispersal model, where the ancestral population originally occurred on mainland South America, and a small subset of that population colonized the Galapagos Islands and subsequently diversified and became recognizably different species than the mainland species. So allopatry is thought to be a pretty common cause of speciation. Look at another example in California, and that comes with Joshua trees. These Joshua trees are a pretty iconic desert species. Most of you are probably familiar with. What you might not know is that there are actually two species of Joshua tree, the Western Joshua tree and the Eastern Joshua tree. And at first glance, they might not appear to be terribly different, but as it turns out, they do have some important distinctive differences. First, their morphological features are not identical. Western Joshua trees almost always form from a trunk, and they branch pretty high off the ground, whereas these Eastern Joshua trees don't have such a distinctive trunk, and they're branching really right off of the ground immediately. And so if you look carefully at these pictures, you can see those differences. Also, these two species of Joshua tree have two completely different obligate pollinating moths, or two different moths that pollinate them, and they depend upon these moths for pollination. So there's one species of moth that pollinates the Western Joshua tree, and a completely different species of moth that pollinates the Eastern Joshua tree. And so this is something else that's driving some important distinctions between these species. But the question is, well, before we get to the question, we can look at this map showing their distribution across the Mojave Desert. The blue represents the Western Joshua trees, and the green represents populations of the Eastern Joshua tree. And we can start asking the question about, well, did these two species diverge via vicariance or dispersal, assuming that they share a common ancestor, which we're pretty confident that they do. And it's hard to say without more information. Did this geographic barrier here that is sort of caused by the lowlands of the Mojave Desert, by the basin of Death Valley, by some geographic separation, cause vicariance? Or did a small subset of either the Western or the Joshua tree disperse across the Mojave Desert from one side to the other and lead to the formation of a new population? Kind of hard to say, but this is the type of thinking that evolutionary biologists have when they're starting to ask questions about, are these things different species, and how did they become different species? The other model we talked about was sympatric speciation, or sympatry. And this basically occurs when resource variation separates populations. And I should point out that sympatry is a fancy term for same environment. So this is speciation that's happening within the same environment as opposed to different environments, as we saw with allopatry, which means different countries or different environments. And so we might have a similar situation where we start off with this original population, but over time, certain individuals in this population are using some resources in the environment more often than they're using other resources, and vice versa. Other individuals in the population are using some completely different resources more often than not compared to the others. And so we have these sort of two subsets within this population that are really utilizing two different kinds of resources in the same environment. And this is causing them to undergo some level of reproductive isolation because individuals using resource A, we'll call it, have a higher chance of mating with other individuals who use resource A because they're all hanging around that resource. Whereas individuals that are using resource B have kind of a higher chance of mating with individuals who are using resource B as well because, again, they have a higher frequency of hanging around that resource. And if that initial step of reproductive isolation starts to unfold, this again can start leading to the accumulation of genetic differences between individuals using resource A and individuals using resource B, and maybe even some different selection pressures. Perhaps there's certain physiological traits that are favored for using resource A and different physiological traits favored for using resource B. And little by little, this is reinforcing the reproductive isolation between these two groups as they're experiencing this thing that we call resource partitioning. And so this is another way that speciation can happen, but it's not happening because of any geographic barriers or anything like that. It's happening more because of resource barriers. And as it turns out, this can happen in relatively small geographic areas. And so what we see is that sympatric speciation is something that tends to be able to happen within a single environment if that environment is really complex and variable, allowing these sort of different ecological opportunities to exist. Resource A, resource B, for example, maybe even resources C, D, E, and F. We can see a real example of this, a famous example, in these cichlid fishes of Lake Apoyo, which is a sort of geologically young lake in Nicaragua. It's only about 23,000 years old since this, it's only been about 23,000 years since this lake was formed. And we see these two different species of cichlids in this lake, the Midas cichlid and the arrow cichlid. And these little pie charts down here at the bottom illustrate the proportion of different resources from the environment that these fishes are using for food. And as we can see, the proportions of these resources that they use are not the same. Uh, Basically, the Midas cichlid is using a lot more algae for food compared to the arrow cichlid, which is instead feeding very heavily on something called biofilm, which, if you don't know what biofilm is, it's basically just layers of bacteria that are sort of piled up on top of one another. You might call it slime, which you might have accumulating in your bathroom. But anyway, we can start asking questions about which of these two species was the progenitor and which one was the derivative when this lake was first colonized by cichlids. And as it turns out, the Midas cichlid occurs in all kinds of other different lakes, but the arrow cichlid only is found in Lake Apoyo. And so biologists seem to think that Midas cichlids originally colonized this lake only to later diversify into two separate species. The Midas cichlid gave rise to a sister taxon, the arrow cichlid, which in sympatry, via sympatric speciation, diverged because it was able to start taking on a lot more biofilm in its diet as a virtue of this thing called resource partitioning. And this led to very strong reproductive isolation between these two species of fish via sympatry. And so, again, we see a situation where this lake has a very sort of complex and variable ecological environment with lots of different kinds of resources that can be used, and different populations of these cichlids have diverged into using different resources in different ways. There are other resources that they can use as well, we see on these pie charts, but the difference between use of algae for food and use of biofilm for food seems to be the most important factor driving their differentiation. And we can talk about this thing called parapatric speciation or parapatry. And this is usually something that occurs along environmental gradients. So environmental gradients are gradients along which some type of an environmental factor is changing. maybe temperature going from low to hot, or sunlight going from very shady to very sunny, or nutrients going from low concentrations to high concentrations, or precipitation going from low precipitation, low rainfall to very high rainfall, sort of in a linear fashion. Or when we talked about local adaptation, we looked at mussels that occur in Long Island Sound. They're responding to an environmental gradient that's related to the concentration of salt in the water, so a salinity gradient. And so along these gradients, we can see different populations starting to diverge. We might start off with some original population again, where there's no gradient, but then a gradient arises for whatever reason. And so we have this environmental gradient of some environmental factors being low here and some being high here, or vice versa. And this is driving differences in the way natural selection is operating because of local adaptations amongst these populations to this gradient. And over time, this will start to drive selection to will start to drive different genes to be selected along this thing we call a genetic cline. So some genes are associated with higher reproductive success at one end of this gradient, whereas maybe some completely different other genes are associated with higher reproductive success at the other end of the gradient, and maybe some intermediate number of them sort of in the middle of the gradient. Again, much like we see with the mussels in Long Island Sound. And eventually, this can start to drive reproductive isolation because individuals with higher reproductive success on this side of the gradient are going to have a higher chance of finding each other and mating. And likewise, we could say the same of individuals on this side of the gradient. And this is something that often becomes associated with this thing we call a hybrid zone. And so we might have this original population sort of diverge into two separate populations, generally speaking, that are more or less adapted to the conditions on either side of this gradient or on either side of this genetic cline. But in the middle, individuals still might be able to mate with each other and form healthy hybrid offspring. And we can look at a real example of this in northern flickers here in the United States. And these birds have two different separate populations on either side of the continent. We have these red-shafted flickers on the western side of North America, the western side of the United States, and we have these yellow-shafted flickers that occupy more of the eastern part of the United States. But in the middle, where these two populations often meet, there's this thing called a hybrid zone, where individual red-shafted flickers might still be able to mate with individual yellow-shafted flickers and form healthy hybrid offspring. But they're still distributed across this environmental gradient going from west to east, where the environment is changing tremendously in a whole multitude of different ways. And that's probably something that drove their speciation via the process of parapatry along this environmental gradient. And we can see, despite the fact that there are distinctive morphological differences between these two bird species, which, if we applied a morphological species concept, would prompt us to say they are separate species, well, they can still form healthy offspring. They don't have reproductive isolation necessarily, other than the fact that they're distributed across geographic space very broadly. Now, this turns out that this plays out to be pretty important for the diversification of birds, because geographic variation really can influence the diversity of different song dialects that occur amongst different bird populations. And song is really, really important for successful bird reproduction. In short, within passerine birds, those are the songbirds, the males will usually use their song to court a female, and the diversity of the song, the quality of the song, is something that's going to influence the success of an individual being able to reproduce successfully, attracting a mate successfully or not. And these male birds usually also have to teach these songs to their male offspring as well. But as it turns out, because not all the environments where certain birds might occur are exactly the same, this kind of changes the sort of acoustic signal that is being sent out in these different places. So we can look at this example from the canyon wren. And we can look at these 6 different populations of canyon wrens and 6 different songs that come from these populations. And as it turns out, 3 of these songs are very, very similar to one another, and the other 3 are more similar to one another. And this is interesting because different geographic distribution of bird songs can be an initial step in speciation, wherein individuals who are singing song type D, let's say females who are hearing song type D sung by males, might not recognize individual males who sing song type B as their own species. And so individual populations that use song type D are going to have a higher chance of mating with one another, whereas individuals who use song type B are going to have a higher chance of mating with one another. And this is almost directly being driven by differences in the geography. Different bird songs will project in different ways. Let's say, for example, song type D might occur in really closed forested habitats. And so that's causing sound to project in a certain way and causing certain sounds to be favorable, certain songs to be favorable, whereas song type B might occur in much more open habitats where the sound's projecting in completely different ways, causing completely different kinds of songs or somewhat different songs to be favored in those environments. And so this is one way that we can see maybe the distribution of songs influencing gene flow in such a way that it's driving reproductive isolation, and perhaps these wrens could divide into separate subspecies, separate subpopulations. Ultimately, maybe if the reproductive isolation is strong enough, they would diverge into 2 separate species. And this is what we call incipient speciation. So it's the early yet sort of incomplete stage of total divergence between lineages. So we have evidence that they are beginning to diverge, but they're not totally reproductively isolated quite yet. And we can see an example of this with plants, with these monkey flowers that occur here in San Diego County. This is in southwestern California. Down here is the U.S.-Mexico border. Here is where the city of San Diego is. Anyway, these monkey flowers have these 3 interesting different forms, these different forms of colored flowers. Along the coast, they tend to have very, very bright red flowers. Sort of going more inland up into the mountains, their flowers are much more orangish. And on the opposite side of the mountains, which is actually sort of becoming the desert at this point, their flowers are much more yellow. This is interesting. These are all the members of the same species as they were circumscribed, yet they have tremendous variation in their floral forms across this environmental gradient. And this gradient, if you don't know, is going from relatively cool and relatively wet to relatively warm and very dry as we get out to the desert from the coast. And also, as it turns out, these populations of the red monkey flowers are pollinated exclusively by hummingbirds, whereas these populations of these yellow monkey flowers are pollinated exclusively by these insects called hawk moths, which surprisingly, these hawk moths are almost the same size as hummingbirds, so they're pretty large insects. Anyway, this is a process where we see this environmental gradient uh, not only varying in terms of temperature and precipitation, but varying in terms of the availability of pollinators. And we think this is something really driving differences in these populations, which may ultimately cause total reproductive isolation, where this one species of monkey flower might ultimately diverge into 3 different species. Now, something very similar to parapatric speciation that we see is the evolution of so-called ring species. And this is something that really depends upon the geographic distribution of populations and the amount of gene flow within them. And so a hypothetical example of this might be a series of populations that are distributed, say, sort of around the edges of a lake in a ring, so to speak, hence the name. And we can see where there are different amounts of gene flow between these different populations. And of course, the populations in closest proximity with one another have the highest chances of exchanging genes compared to populations that are not very geographically close to one another. And a real example of this we see comes with these Ensatina salamanders here in California, who form a ring species sort of along the Pacific coast and then all the way down through the Sierra Nevada mountains around the San Joaquin Valley. And as you might notice, there's this little gap right here or these populations are not exchanging genes. And as it turns out, we have these 7 different subspecies of Encetina distributed around this ring or along this ring. And some of these populations might be able to exchange genes within certain hybrid zones that exist. But some of these populations have a very, very small chance of exchanging genes with one another. So once again, we see this situation where uh, large scales of environmental variation going from Southern California to Northern California. That's, again, sort of a gradient of warmer, drier conditions to cooler, wetter conditions going from Southern California to the north. And likewise, we see something similar in the Sierra Nevada mountains are probably going to be cooler and wetter than just about anywhere else. And this has caused natural selection and probably some level of genetic drift to cause differences in these different species to the point where experts who study salamanders have recognized that, hey, these things are really different subspecies within this genus, and we should recognize them as such, but they are still capable of hybridization. And so we might ask questions about what's causing these differences. Is it really because of natural selection? Is it really because of genetic drift? Or are both things sort of operating at the same time? It's kind of hard to tell at face value, but these are the kinds of questions that evolutionary biologists would ask. And this leads to the concept of something we call secondary contact. And secondary contacts, basically the situation where a population splits into 2 separate populations or more, but let's just say for the sake of explanation, 2 separate populations, and there's some barrier between them causing this separation, and there's some level of divergence, probably genetic divergence, and barriers to dispersal, barriers to them sharing genes. But then all of a sudden, these 2 populations come back together. And this happens in nature. And then there are basically 2 possible outcomes via this process of secondary contact. There will be some hybrid matings between the 2 individuals from the 2 separate populations. But the question is, do the hybrid offspring have high relative fitness or low relative fitness? If they have low relative fitness, secondary contact will reinforce the process of speciation. If these individuals from these 2 populations come together, they're making offspring, but those offspring are not very viable, not very fertile, probably not going to have offspring of their own. Again, we could apply something like the biological species concept and say, via secondary contact, we know that these things really are sort of like 2 different species. In contrast, what if these hybrids, or some of them anyway, tend to have really high fitness? Well, this would sort of go against this idea of reinforcement. And this might allow this hybrid population to persist and lead us to determine that, hey, you know what? Even though there's been divergence between these 2 populations, or divergence between these 2 populations, and some level of reproductive isolation between them for presumably long periods of time, they can still come back together and breed and produce perfectly healthy offspring. And that is kind of what we're seeing going on with these ring species, at least the populations of them that are sort of distributed closest to one another. There's not strong reproductive isolation between them. And so hybrids can form. And hybridization via this thing we call secondary contact can really lead to the formation of new species. And so we can kind of look at some different factors here: hybrid fitness, the characteristics of a hybrid zone, and then we can kind of look at what the potential outcomes of these hybrids are, of these hybrid offspring are. And so if the fitness of the hybrids is lower than that of the parent species, and a hybrid zone is really sort of small, that is to say, narrow, short-lived, but a small hybrid zone, probably the parent species will become even more different from one another over time via this process of reinforcement, which we see here in this cartoon. If instead the fitness of the hybrids is equal to that of the parent species, and the hybrid zone is sort of large and long-lived, like we saw in our example here with northern flickers, then perhaps we'll see a situation where the parent species are going to coalesce. And so, despite the fact that there's geographic separation, perhaps some level of reproductive isolation might exist, but these two populations still produce perfectly healthy offspring, then they're really kind of going to be still considered a single species. However, if the fitness of the hybrids turns out to be higher than that of the parent species, and it happens in something we call an ecotone, which is basically where two different kinds of habitats overlap, like a shrubland overlapping with a forest, or if this even happens in a whole entirely new habitat, we could either see again a stable hybrid zone, but particularly in the context of this happening in a new habitat, we could see the formation of a new species. And the reason why we think of this as being possible if the hybrid zone, or if this happens in a new habitat, I should say, is that maybe the hybrids have certain traits that are better adapted to the new habitat compared to the parent species. And this can really lead to them going off to form a whole new lineage that we could ultimately end up considering a separate species. This whole business of hybridization gets kind of crazy because there can be a lot of back crossing between parent species and the hybrid offspring. And this causes something we call a hybrid swarm to form. An example of this comes with these cottonwood trees. And we have these two different species of cottonwood, Populus fremontii and Populus angustifolia. And this map here kind of illustrates an area where these two species of cottonwood have separate populations, but there's an area where their ranges overlap. And in these areas where their ranges overlap, they form hybrid offspring. Now, these hybrid offspring can mate with either of the two parent species. And so we have matings between the hybrids and the two different parents sort of going back and forth, to the point that it kind of becomes like this species complex, where one species is kind of indistinct from the other at a certain point in time because they've formed so many hybrid offspring, and those hybrid offspring have also mated with the parent species quite a bit. And this can make species really sort of indistinct. Like, where does one species start and the other one and end, and where does the other one start and the other one end? It turns out in this case, it's kind of vague. An example is seen in the different shapes of the leaves that are found in this particular environment, where some individuals produce leaves that are very much characteristic of Populus fremontii, and some individuals produce leaves that are very characteristic of Populus angustifolia. But in the hybrid zone, there are many different individuals carrying leaves of intermediate shape. And this is a direct result of all this hybrid back crossing going back and forth. So this does happen in nature, and it really very much throws a wrench in our concept of what a species is. Is this a single species? Well, evolutionary biologists probably would again call this a species complex, where in certain areas they're very distinct from one another, but in other areas, because of so much extensive hybridization, they are really indistinct from one another. As it turns out, sometimes hybridization can actually have negative consequences. And this can happen via the process we call introgression, genetic introgression, often happening within hybrid swarms or even just between hybrid mating events. And this can lead to this thing we call gene swamping. And a classic example of this comes with these tiger salamanders. And we have a species of tiger salamander here in California, which is an endangered species. As it turns out, about 50 years ago or so, during the 20th century, a non-native species of salamander called the barred salamander was introduced to California for fishing bait. And reproductive isolation between California tiger salamanders and these Texas barred salamanders is almost non-existent. It's very, very weak. They can produce very viable hybrid offspring. And in fact, as it turns out, their hybrid offspring have higher fitness than either of the two parent species. And these hybrid offspring have become very, very, very prevalent in the environment, to the point where the hybrid offspring are outcompeting the endangered parent species, the California tiger salamander. And this might potentially threaten this endangered species because the genome of the California tiger salamander, the full complement of genes that make up the genome of this native salamander is being contaminated, if you will, by the genome of this Texas barred salamander. And so these hybrids aren't pure California tiger salamanders. Their genomes are not purely composed of the same DNA sequences as the California tiger salamanders, many of which were their parents. They're instead a mix of genes from the California tiger salamanders and the barred salamanders. And if these hybrids continue to have high fitness for many, 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 many generations, they'll basically be the lineage that persists. And much of the genome of the California tiger salamander may be lost via the process of sexual reproduction, genes getting mixed up during sexual reproduction, and certain California tiger salamander genes that might be important for that species could be lost. And so this is, again, what we call gene swamping. The genome of this native endangered California tiger salamander is being swamped out by the genome of this non-native introduced Texas barred salamander. And this has important conservation consequences because there is probably going to be some loss in the genetic diversity of the California tiger salamander. Another sort of weird and unintended consequence of hybridization can be seen in Darwin's finches. And sometimes, as it turns out, juvenile birds are abandoned. This is the nest of a cactus finch. And sometimes, or this is the nest, this is a nest of Geospiza scandens, I guess I should say. But sometimes when these nests are abandoned, a different species will take up this nest and raise the offspring. And we see examples of that happening between Geospiza scandens and Geospiza fortis. And as I mentioned before, these birds, which are passerine songbirds, use song to attract their mates. And the male birds are the ones doing the singing, and the male birds learn their songs from their fathers. And so if this foster offspring learns the song of a parent species that's a different species, then it might attract a mate of that species and not a mate of its own species. And it might be able to successfully mate in doing so, but it's going to form a hybrid offspring in the process. And we can start asking questions about how might this affect the fitness of the hybrid offspring? What song is the hybrid offspring going to learn? And so this is just another way that hybridization happens, and it can lead to some sort of complicated and unintended consequences. And this whole idea of hybridization points to the idea that when we think about phylogenies, evolutionary trees, we think of them as being very much composed of linear branches, linear branching patterns. One ancestor gives rise to two branches, and then another ancestor gives rise to two more, and so on and so on and so forth. But when we start thinking about hybridization, we come to the realization that not all phylogenetic relationships necessarily do involve linear branching patterns. We can see this even in the early evolution of life, maybe not so much because of hybridization, but maybe because of this similar process that we call horizontal gene transfer, where more primitive, often single-celled life forms that existed long ago have been exchanging genes sort of accidentally back and forth from one lineage to the next. This might have happened with things like the mitochondria or things like chloroplasts, which are both organelles involved in organelles that are pretty essential to many eukaryotic life forms. And so there's this sort of braided pattern of phylogenetic relationships that unfolds as a result of this. And we see the same thing with hybridization, where we might have these 5 lineages of sunflowers, but all this back crossing via hybridization during the process of secondary contact really leads to sort of, again, this more braided phylogenetic pattern where these species aren't necessarily branching off in these linear ways. They're back crossing with one another to the point where the tree is more kind of like a bush. So anyway, this is just a realistic way that different processes of speciation play out. We like to think of things as being very neat, and we like to put things in boxes and build our human constructs, but nature operates in its own ways sometimes. and that makes our interpretation of it a little bit more challenging. So just to reiterate, we see the situation here where there are sunflowers, which by the way are in the genus Helianthus, which is what the H stands for, where two different species have come together to form hybrids. For example, Helianthus petiolaris and Helianthus annuus have come together to form Helianthus anomalus, or Helianthus petiolaris, and maybe Helianthus annuus could come together to form Helianthus paradoxus. And so this is just an example of this braided pattern that unfolds when we have hybridization events. And again, to mention, each of these different hybrids that are formed by different types of sunflowers occurs in sort of different habitats, perhaps compared to their parent species, where they can do really, really well. They could do better than their parent species because of certain attributes of hybridization. So hybridization has a lot of interesting outcomes. It can be positive, it can be negative, whatever the case may be, it certainly exists. And probably one of the most bizarre ways that we see the process of speciation playing out, often in the context of hybridization, is via this process we call polyploidization, which is a little bit far out there, and I don't want you to get overwhelmed by it, but I invite you to recognize that the word polyploidization refers to the fact that this involves situations where the cells of individuals carry more than two sets of chromosomes. So when we say haploid, we're talking about a cell that has one set of chromosomes. When we say diploid, we're talking about cells that carry two sets of chromosomes. When we say polyploid, now we're talking about cells that carry many extra sets of chromosomes. And this has happened a lot in plants, and particularly in the mustard family, or known as the Brassicaceae. These are all mustards, and actually many of these mustards might be familiar to you. You may have probably eaten some of them before. But diversification within the mustard family has very much unfolded via the process of polyploidization. How does this happen? In short, during cell division, there's this process called meiosis. And during meiosis, this is when cells are dividing to form gametes. And typically during meiosis, a diploid cell will divide into 4 daughter haploid cells. But sometimes there are mistakes, and that doesn't happen. And instead, sometimes this diploid cell doesn't divide, and instead it forms 2 diploid daughter cells and 2 daughter cells that don't have any chromosomes at all, that are not going to be able to be involved in fertilization. But now we have a situation where the gametes are diploid, when ordinarily we would expect gametes to be haploid. And if these diploid gametes come together during fertilization, so if this mistake happens in a male, let's say, for example, a male pollen grain, and the same kind of mistake happens in a female, say a female egg, where now the male pollen grain is carrying twice the normal number of chromosomes, and likewise, so is the female egg, and that pollen grain fertilizes that egg. Now the offspring is going to have twice the normal number of chromosomes. And so instead of forming a diploid offspring as we would normally expect, we now have a tetraploid offspring formed. And by virtue of carrying different numbers of sets of chromosomes, we would call this offspring a new species. And so we could see examples of Brassica nigra undergoing these mistakes. And we could see examples of Brassica oleracea undergoing these mistakes. And they're actually 2 different species who have undergone these mistakes, forming a hybrid offspring with twice the number of normal sets of chromosomes. And this has also happened between Brassica nigra and Brassica rapa to form Brassica juncea, and between Brassica oleracea and Brassica rapa to form Brassica napus. And so these hybrids, by virtue of carrying double the normal set of chromosomes, are able to undergo hybridization and form offspring that have, again, twice the normal number of set of chromosomes that you might expect, and really form completely new species. So Brassica carinata is a new species formed by the hybridization of Brassica nigra and Brassica oleracea, for example. And we can say the same thing about the hybridization between these other pairs. And this has happened not just in the mustard family, but in all kinds of other domestic plants, tobacco, wheat, oats, sugarcane, cotton, et cetera, et cetera. And as it turns out, polyploidization has been pretty beneficial in the formation of domestic crops that are useful for us. And oftentimes, by virtue of carrying twice the normal number of chromosome sets, these individual hybrids produce larger morphological forms. So certain characteristics that we might favor for our own uses for these things as food are favored by the fact that they are polyploids. And so they're kind of growing larger than they might have, than either of the parent species might have, I should say. So these hybrid offspring are oftentimes kind of going to be larger than than the parent species. Something similar we also can see in whiptail lizards. And this is something that rather I should say, this process by which two different species come together to form a new species via hybridization under polyploidization is specifically called allopolyploidization. Don't get too lost in the technicality of the terms. But again, we have a situation where this one Nematophorus species has formed polyploid gametes by accident, and this other Nematophorus species has also formed polyploid gametes by accident. And these two species have come together to mate and form a hybrid offspring, which is Nematophorus neomexicanus. And this is a polyploid species, a new species that has been formed by the mating between two different species that each just happen to carry polyploid gametes by chance alone. This is really just happening by accident and forming a whole new species. And as it turns out, polyploidization can benefit sterile hybrids, and it can allow sterile hybrids to become fertile. So if we think about two species crossing, and so this species here carries this particular set of chromosomes, which are coded by color, and this particular species here carries a completely different set of chromosomes coded by completely different colors, and they cross, they'll form this sterile offspring. And the offspring is sterile because they do not have the proper complement of homologous chromosomes. However, if this individual undergoes polyploidization, that is, mistakes during meiosis that lead to gametes containing twice the number of normal chromosomes, and if that happens in another individual, whether or not that individual is of the same species, we will now have a situation where there is a proper complement of chromosomes because of these accidents caused by polyploidization. And this can allow the offspring of a hybrid mating to become fertile. And if this happens to multiple hybrid offspring in a population, those hybrid offspring can come together and produce their own offspring that will presumably also be fertile. And so polyploidization has been a pretty effective way of allowing sterile hybrids to become fertile. And overwhelmingly, we have seen this happen in plants. And this has been probably really, really important for plant diversification, particularly in what we call lower plants, or plants that diverged in evolutionary history a long time ago compared to those that diverged more recently, particularly ferns. We can see these different ferns here that have, and lycophytes as well, that have pretty high proportions of species that were formed via polyploidization and have really, really high chromosome numbers. And we can see that in some flowering plants as well. But this process of accidents during meiosis, accidents during cell division, and the formation of gametes that carry extra sets of chromosomes, for some reason, seems to have become much more prevalent in plants than any other taxonomic group. We do see an example with the Nematophorus lizards. By and large, this is mostly something that we see in plants, and it is something that is thought to have really tremendously contributed to the diversification of plants, and especially not just these lower plants, but some of our domestic crops, as we see with mustards and some of these other agricultural crops that are probably pretty familiar. So anyway, this is in a nutshell kind of a complicated way of how we understand what species are and how it is that we go from having one species to having multiple species that diverge in many, many lineages. And to understand it is sort of necessarily complicated, but that's just kind of the nature of the beast. Life is complicated, and evolutionary diversification simply just is just not a simple process. It's kind of inherently complex. All right, so that's it for this lecture. I'll catch up with you next time.